Today we will first learn the organization of the skeletal muscle. Skeletal muscle contains elongated, thread-like multinucleated cells called muscle fibers. The most striking feature of skeletal muscle fiber are an orderly arrangement of contractile myofilaments into myofibrils and a characteristic stretching pattern. Each myofibril has alternating light and dark bands along its length. Two sets of myofilaments, thick and thin, that make up each myofibril are organized into repeating units of contraction known as sarcomeres. Dark transverse Z bands mark the ends of each sarcomere and arch the thin filaments. The center of the sarcomere contains the thick myosin-containing filaments, which form the A band, thin actin-containing filaments, which form the 1 band, are at the end of each sarcomeres. This is the longitudinal section of the skeletal muscle. The muscle fibers are elongated cells grouped into fascicles, bundles. Each muscle fiber displays a corresponding pattern along its length. Flattened nuclei are located directly under the sarcomere of each cell. Here is the transverse section of the skeletal muscle. The muscle fibers are rounded to pleomorphic cells with peripheral located nuclei. The surrounding perimysium contains an extensive network of capillaries and small blood vessels. Then we will learn the major components of skeletal muscle fibers. The sarcoplasma is the plasma membrane of the muscle fiber, enveloped by an outer layer of glycoproteins and a thin network of reticular fibers, all constituting an external lamina. The sarcolemma and its tubular invaginations, called transverse tubules, play a role in the initiation of contraction. The myofibril constitute about 18% of the cell volume. Each myofibril contains a closely packed, orderly, overlapping, longitudinally arranged array of myofilaments with a highly regular binding pattern. Each sarcomere of a myofibril consists of a band and half of two continuous 1 band. The sarcotubule system is composed of two separate and distinct membrane systems of the muscle fiber, known as the sarcoplasma reticulum and the transverse tubular system. This is the segment of the skeletal muscle fiber, enlarged to show its major components. Here is the details of the muscle tride. A transverse tubule invaginates from sarcolemma to closely associated with the terminal cisternae of the SR to form tride. The third part is the histology and ultrastructure of skeletal muscle fibers in longitudinal section. The skeletal muscle fiber constitutes 14% to 15% of the body weight. As its name implies, it is mostly attached to the skeleton. It is also called striated or voluntary muscle because its cells appear striated or correspond under the microscopy. Skeletal muscle fibers are elongated cylindrical cells with tapered ends. They are multinucleated, the nuclei occupying peripheral positions in the cell. Their cytoplasm, known as sarcoplasm, is packed with myofibrils, which are cylindrical bundles of myofilaments along the length of the fiber. Each myofibril has a uniform diameter and consists of identical repeating units called sarcomeres. Sarcomeres are composed of longitudinally arranged thick and thin filaments and perpendicular Z-bonds. In longitudinal section, skeletal muscle fibers show transverse striation because adjacent myofibrils are in lateral register with each other across the width of the fiber. The greater density of A bonds is due mainly to the presence of thick filaments, whereas the light density of one band is due to the prevalence of thin filaments. In the center of each A band is a light H zone, the central part of which filaments not overlapped by thin filaments, which is bisectioned by a thin dark M bond. Here is the picture of light micrography of part of skeletal muscle fiber in the longitudinal section. The regular cross striation pattern is clear. Alternating dark and light segments cross the fiber from one side to the other. The dark stripes correspond to A bond of myofibrils, whereas the light stripes are the one bond, bisectioned by very thin C bonds. Several elongated nuclei are located in the peripheral areas of the cell. The surrounding loose connective tissue constitutes the endomysium, which contains nuclei of scanted fibroblast. The fourth part is the histology and structure of the skeletal muscle fiber in transverse section. In transverse section, stained with hemotoxin and eosin, the cytoplasm of each fiber is intensely eosinophilic and looks punctate because of tightly packed myofibrils. Myofibrils constitute the bulk of each fiber and mostly contain the major contractile proteins, myosin and actin, which make up the myofilaments. In cross-section, myofibrils are often seen grounded together and form irregularly shaped coherent field. Here is the picture of light micrography of skeletal muscle showing parts of several muscle fibers in transverse section. Eosinophilic staining characteristic and punctate appearance are due to the contractile proteins, which constitute much of the cytoplasm of each cell. Multiple rounded to oval nuclei sit under the cytoplasm of each cell. Nuclei of statellite cells are not visible in conventional hemotoxin and eosin sections, but they lie between the sarcolemma and the thin external lamina. Nerve fascicles of various sizes and capillaries abound in surrounding endomysium. Finally, we will learn the sarcomere and myofilaments in contraction. The global head or its one region contains an ATP paste that facilitates bind to actin to move to the head and produce a powerful stroke. Antiparallel association of myosin molecules form thick filaments. In half of the thick filament, myosin heads are oriented in one direction. Those in the other half are in the opposite direction. The tails of myosin molecules overlap, which yields a bare central shaft. Two proteins associated with actin, tropomyosin and troponin complex, respond to varying calcium ions concentration by actin as a switch to enable or disable the interaction and the formation of the cross bridge between actin and the myosin heads. Myosin heads bind to actin and join the thin filaments a short distance apart the thick filaments. Thin linkage break and reform further along the thin filament to repeat the process. Filaments are thus paired past each other in a ratchet-like action. This picture shows the muscle contraction and relaxation. Interdigitation of thick and thin filaments allows sarcomere contraction, which is best explained by the sliding filaments model in which actin filaments slide along myosin filaments. During contraction, both sets of filaments retain their normal length. 8 bands remain unchanged in length. 1 band shorten, and each zones are narrowed.